Good morning, students. I'm Dr. Sumitra My chapter today is about the general introduction to parasitology, and then there is another one more topic which is called Entamoeba histolytica. Okay. So first of all, before going to Entamoeba histolytica, let us know there will be this general introduction to parasitology. Okay. So let us know what is parasite. So by parasite, we mean parasite is a living organism which lives in or upon another organism, which is called as a host, and derives its nutrients directly from it without giving any benefit to the host, which means parasite is a living organism. It should be living, and so in order to thrive or in order to live, they have to depend on another organism which is living. Okay, and this is called as the host, and they derive all the nutrients directly from it, but the thing is that it is not benefiting to the host. Okay, so in medical parasitology, we are dealing mostly of protozoa and helminths. So helminths are the one which is called as the animal parasite. So basically we are animals, so we are dealing mostly with helminths. Then parasite, we already know parasite are those organisms which thrive mainly on the living organism for their living, right? So we have again divided this parasite into two types. There is the ectoparasite and endoparasite. By ectoparasite, we mean ecto means outside and endo means inside. Okay, so the definition for ectoparasite is very important. It is the parasite that inhibits the surface of the body of the host without penetrating into the tissue. Example, the flea or ticks. They are important vectors transmitting the pathogenic microbe. The infection by this parasite is called as the infestation. So by ectoparasite, it means ectoparasite, it's largely thrive on the surface of the body. They never penetrate they never penetrate inside the body. Okay, so they are this infection which is caused by the ectoparasite are called as the infestation. By endoparasite, we already know. So ecto is outside, so endo will be inside only. It is the parasite which lives within the body of the host. Example like leishmania. Invasion by the endoparasite is called as the infection. By ectoparasite, we call this as the infestation, and by endoparasite, we call it as the infection. So, and then this endoparasite, mostly we are going to deal about endoparasite, okay, because endoparasite are the one which mostly cause infection to us, right? Endoparasite are of the following types. They are obligate, facultative, accidental, and aberrant or the wandering parasite. Then obligate parasite means this is a parasite which cannot exist without a parasitic life in the host, means they need a host for completing their life cycle. This is called as the obligate parasite, like Plasmodium species. Plasmodium species are the one which causes malaria. They are obligate parasite. Okay. Facultative parasite means this is the parasite which can live a parasitic life or free living life when the opportunity arises. Means they can either live with the host or without the host. For like a amoeba. Okay, and then accidental parasite. Accidental parasite are this is the parasite which infects an unusual host. Example, Echinococcus granulosus infects the human accidental. Okay, then aberrant parasite or wandering parasite. This is the parasite which infects a host where it cannot live or develop further, like Toxocara canis and Toxocara cathy in humans. Okay. Then see, what do you mean by host? Host means we the human beings or anything. It may be us, it may be animals or anyone. Okay, but the thing is that the parasite has to come and they have to infect us. This is the host. So by host, we mean host is defined as an organism which harbors the parasite and provide nourishment and shelter. So suppose if we are the host, okay, then let's for example Plasmodium, that is the mosquito. Mosquito is a parasite. Why? Because they used to come and they used to infect us, and we are the host. Okay, so that means we are giving nourishment to them, right? So we are the host. That is the example of a host. Okay, so host may be again of two types. Okay, definitive host and intermediate host. Definitive host means a sexual cycle. Sexual cycle means where reproduction will be there. For example, like Anopheles species for the Plasmodium. And intermediate host is the one where the asexual cycle is being take place. For example, men for Plasmodium. Okay. Then host can also be the reservoir host, paratenic host, and amplifier host. Reservoir host means this one. Reservoir means what? Reserve, where reservation is there, where we harbor the parasite. And they serve as an important source of infection to other susceptible hosts. Means, for example, parasite will come and they will infect the the host, okay, and they will reserve the host so that they can infect the other people or the other host, okay. This is called as a reservoir host. Paratenic host means it is the host in which the parasite lives, but it cannot develop further and not essential for its life cycle. The difference between reservoir host and paratenic host is that by reservoir host, they will infect the other organism. By paratenic host, they won't infect the organism, they will just be within themselves. This is called as the paratenic host. Freshwater prawn and crab for the Angiostrongrellus cantonensis, big suitable fish, etc. Amplifier host. What do you mean by amplifier host? By amplifier host, we means the parasite will live and multiply exponentially. By reservoir host, it's only for a temporary place. They can infect the other organism. Paratenic means they won't infect the other organism, they will just live inside it. And amplifier host means they will multiply it. That is the difference between the three hosts, okay? This is very important, you mark. Then host parasite relationship. See, not all the parasite are like what to say are very bad to us. Okay, some is like some relationship are there. Okay, they are called as symbiosis, commensalism, and parasitism. By symbiosis, what do you mean by the symbiosis? It is the close association between the host and the parasite. Both are interdependent upon each other that one cannot live without the help of the other. That means that uh, by symbiosis, we mean that there should be a cross relationship between the host and the parasite. That means the host they cannot live without the parasite, and the parasite they cannot live without the host. That means like couples type. Okay, so by symbiosis, we means they are couples. Okay, just remember like that. So that means the host is the boy and the parasite is the girl. So they are dependent on each other and they cannot live without each other. So they are dependent to each other. Okay, this is called symbiosis. By commensalism, which what? It is the association in which the parasite only derives the benefit without causing any injury to the host. This is called as a commensalism. By symbiosis, this means both the girl and the boy, they cannot live without each other. That is a symbiosis. Commensalism means what? Either the girl or either the boy, they will benefit everything from the other. For example, like the parasite is a girl and the boy is the host. Okay, the parasite will derive everything from the host. That is, the girl will derive everything, all the benefit from the boy. Okay, this is called commensalism. Okay, then parasitism is what? It is an association in which the parasite derives benefit from the host and always causes some injury to the host. Okay, the host will not get any benefit in return. Means the girl, okay, that is the parasite. Okay, the girl that is the parasite, they will benefit everything from the host and they will leave the host. That means the boy, and then the boy will die of this one, will be ended up in heartbreak. Sorry, this is called injury. Okay, symbiosis means they cannot live without each other. Commensalism means the parasite will derive everything from the host, but they cannot give any heartbreak to the host. That is the boy. Okay, parasitism means the parasite will derive everything from the host. Okay, and in return the host will be left with this one, broken heart. Okay, this is the injury. You got the point now?
Yes, ma'am.
Ma'am.
Okay,
Yes, ma'am.
okay. Now let's go
Yes, ma'am.
transmission of parasite. Okay, see, transmission of parasite, it depends upon source or reservoir of the infection and the mode of transmission. See, source of infection, it may be human beings like us, then animals like what to say क्या बोलता है these dogs, then cats and all. Okay, then vectors means the carrier. Okay, anopheles for malaria, Culex for filariasis, sandfly for Kala ajar, reed bug for the Chagas disease, and sassafly for the sleeping sickness. Then contaminated soil and water, like soil for like hookworm, Ascaris, Trichuris, water like Entamoeba histologica or Giardia lambia. Okay, then raw or undercooked meat. Why cooking of food is very necessary? Cooking of food is very necessary. Why? Because they contain some parasite like Cysticercus bovis and like Cysticercus cellulosis. Okay, which is very bad to our very bad to our health. So for this reason, we have to eat cooked food. Then mode of transmission, this is the source of infection. Okay, then the mode of transmission, how they are transmitted. It can be transmitted from oral or fecal-oral route. Oral means by eating, then fecal-oral means that everyone will know through like this thing, feces and all. Okay, then penetration of the skin and mucous membrane, this will be for the ectoparasite. Then when it penetrates inside the skin, it will become the endoparasite. By sexual contact like Trichomonas vaginalis, bite of vectors like mosquito, for example Anopheles. Okay, see, Anopheles is the vector and malaria is the disease which is cause. Okay, vertical transmission means from mother to child. Blood transfusion like Plasmodium, Babesia, Toxoplasma, Leishmania, and Trypanosoma. Then what do you mean by auto-infection? This is very important, okay, in the exam point of view for all of you. Then contaminated hand, like external auto-infection, or by reverse paracelsus, which is called as the internal auto-infection. Auto-infection means what? Auto-infection means infecting one by themselves, okay. Supposing we don't wash our hand, what will be happen? Obviously, we will be eating something, germs or something, and then we are infecting by ourselves, right? So this is called as the auto-infection. And then, for example of auto-infection are Cryptosporidium, Hymelopsis nana, Entropius vermicularis, Strongyloides stercolis, and Tinea solium. Okay. Then life cycle of the parasite. By life cycle of the parasite, we means direct or simple life cycle, then indirect or complex life cycle. By direct or life cycles, what do you mean? When a parasite requires only one host to complete its development, this is called direct. Direct, okay. That means, see, direct and indirect, okay. I will give you an example. By direct one means the parasite, just imagine example is a girl, okay, so they need only one boyfriend, okay, in order to be content or be happy. This is called as the direct or simple life cycle. By indirect or complete life cycle means what? This girl, that is the parasite, okay, they are very bad, okay, they need more than one boyfriend or they need multiple boyfriend. This is called as the indirect or complex life cycle. So it's like simple means like simple relationship, just remember, just think like that. And complex means uska life बहुत complex है this parasite का, ठीक है. So direct means they require only one host, okay, and indirect means they require more than one host. They can be like 2 hosts, 3 hosts and all, okay. This is called as the indirect life cycle. So direct or simple life cycle. Direct or simple life cycle means parasite, they need only one host, that is the man, okay. So it will be like protozoa helminths. Thus you have to know all the examples, okay. Do not all the example. If they ask them by give example of simple life cycle or direct life cycle, just say 1 2 each, okay, like 3 4. Do you have to know the name, okay. This is important. And the indirect or a complex life cycle, okay, which requires more than one host, okay. This is the this one, indirect or the complex life cycle. We have to know the examples, okay. The definitive host and intermediate host. By definitive host we mean what? Sexual cycle has take place, okay, where the host. Intermediate means asexual cycle. This is called as the intermediate host. Then indirect or complex life cycle. See, Plasmodium. Plasmodium is the parasite and definitive host is the Anopheles mosquito, which means the sexual cycle has been taken place in the mosquito. And intermediate host, which means the man, that is the asexual cycle has been taken place in the man. This is the intermediate host. Then you have to know all this example. All this example means here you have to know only Plasmodium is important. Echinococcus granulosus, Toxoplasma, Tinea solium, and then this one, Fasciolosis hepatica and all, okay. Fasciolosis hepatica is important and Paragonymous species more, okay. Then suppose if there is a parasitic infection or parasitic disease, we have to know now how to diagnose this parasitic disease, right? So laboratory diagnosis of the parasitic disease means parasitic diagnosis we can either diagnose either microscopically or macroscopically. Microscopically means we have a microscope, and macroscopically means just by looking, okay, sometimes na. And then culture method, then immune diagnostic method like antibody detection, intradermal skin test, okay, molecular method, genodiagnostic, animal inoculation, imaging technique. Laboratory diagnosis of parasitic disease is important, okay, in your exam, from your exam point of view. Then suppose if there is any we have to know how to diagnose. Then obviously if there is any parasitic disease we have to know now how to treat only. So the treatment for the this parasitic disease, antiparasitic drugs like various chemotherapeutic agents are used for the treatment and the prophylaxis of parasitic infection, surgical management, semi-conservative surgery and all, okay. And the drugs, this is important. Drugs for amoebiasis means amoeba. So metronidazole, tinidazole, and ornidazole. Okay, this is the azole family more. Then mechanism of action is it's not that important. Just know what are some of the drugs for this thing, antiparasitic drugs. Na? Okay, then these are the drugs. We have to know some only, not much, not all. Okay, then This artemisinin derivative, artemisinin or artemether or arteether. Okay, these are very important because they are the drug of choice for complicated or the falciparum malaria. Okay, so they might ask you like in your viva and all, what is the drug of choice for complicated or falciparum malaria? You just have to say artemisinin, like artemisir, arteether, artemisinin and all. Okay, then quinine is important, but in some this one in some person or some people they are not indicated. Why? Because quinine they causes hypoglycemia, so for this reason for they are not indicated. Okay, then uske baad mefloquine, primaquine and all. Okay, lumefantrine and all. Okay, then babesiosis, this is not important, you will just know about the drugs for malaria. Then toxoplasmosis, that is the Toxoplasma gondii. Okay, 4-trimethazole, that is the trimethoprim, sulfamethoxazole. Then this is HIV infected people for drug of choice, which is very important. Spiramycin, drug of choice in pregnancy. Suppose if a person is pregnant or if a girl is pregnant, what to do? We have to give spiramycin, right? So because this is the drug of choice in the pregnancy. Okay, these are important. Then trematodes, cystode, trematodes, and the nematodes and all. So for cystode, you have to know this one: praziquantel, niclosamide, and albendazole. Albendazole is important. Cysticercosis, like cysticercosis and hydatiditis. And albendazole is mostly indicated for children also from for when they have these strong worms and all. Na? For this reason, okay, albendazole. And trematodes like praziquantel. Triclabendazole, it's okay, but praziquantel mostly we give only. Then this is a nematodes and Firia nematodes. Okay, so these are the introduction for the parasitology. Okay, so we will go to entamoeba histolactica. So entamoeba histolactica, this is let's know some history about entamoeba histolactica. So entamoeba histolactica was first described by Fyodor Lodge in 1875 from Russia. The species name was coined by Fritz Saudin in 1903. This is the history, brief history about entamoeba histolactica. So, why this entamoeba histolactica is important? Entamoeba histolactica is important. Why? Because it is the major health problem worldwide. Okay, so the largest burden of the disease, that is the entamoeba histolactica, occurs in tropics of like China, Central and South America, and India also, mostly India. Okay, they affect more than 10% of the world population. Third most common parasitic death in the world after malaria and the schistosomiasis. So malaria it ranked first, which has been followed by the schistosomiasis, which is caused by the schistosoma and all. Then globally, approximately 50 million cases and 110,000 deaths have been reported annually by WHO. The incidence of the invasive disease is considerably higher in urban areas of Mexico City and all. Okay, so see, contributing factors. Which are the contributing factors for amoebiasis? They are poor nutrition, tropical climate, stress, altered bacterial flora in the colon, traumatic injuries to the intestinal mucosa, decreased immunity, alcoholism, and the genetic factors. In India, the prevalence rate is around 15%. It has been reported throughout the India. Okay, so taxonomic classification of amoeba, that is the entamoeba histolactica. So in which class this one phylum it belongs to amoebosa, and then order it this one. This is not at all important. Just remember, not all amoeba belong to kingdom protozoa and subkingdom neozoa. Okay. Then C. Morphology. Morphology of entamoeba histolactica. C. Trophozoid. Trophozoid, pre-cis and cis. Cis is the egg form, okay? And then the trophozoid form is the adult form. See, in that this one, in the here, this is a pseudopodium. Why pseudopodium is required? Pseudopodium is required for movement. Okay, they move through with the help of the pseudopodia. Okay, this is a pseudopodium. Ectoplasm means which lies outside, and this is the WBC. Endoplasm is the inside one, which is filled with the fluids. Okay, and this is the karyosome. Karyosome is the nucleus. Then peripheral chromatin and all. Then RBC. Then food vacuole. Food vacuole is the one where they will trap the food. Okay, okay. And this is the pre-cis. Okay, cis is the egg form. Okay, egg form. See here in the ectoplasm is there, endoplasm is there, chromatin network and all formation are there. Here we don't see any RBC or WBC. Then in the uninucleated cis means two nucleus formation will be there. Okay, here see. And this is the glycogen mass and all. Then binucleate and the quadrinucleate. By binucleate we have two nucleus, and then you quadrinucleate we have 1, 2, 3, 4 nucleus. Okay. These are the morphology of the entamoeba histolactica. This is important, huh? Then Next to life cycle. See, host. Entamoeba histolytica completes its life cycle in the single host, that is the man. So what do we call one which completes its life cycle in single host? You know. What do you call when they complete their life cycle in the single host?
Ma'am, monochromatic life
Ma'am.
cycle.
Simple, ma'am.
Simple, correct. Direct or simple, okay. So see this entamoeba histolytica, they complete its life cycle in the single host. That is the only one host, and that is the man, okay. Then infective form, that is the quadrinucleus cyst. Quadrinucleus cyst, we already know. This is the one. This is the infective host, okay. 1, 2, 3, 4. This is the one which will infect the host, okay. And then mature quadrinucleated cyst is the infective form. However, uni or binucleated cyst can also be infective, but mostly the quadrinucleated cyst. Okay. Mode of transmission is with the help of the fecal-oral, that is with the help of through feces and all, or through sexual contact, or through any vector. Okay. So let's know about what are the virulence factors. Okay, virulence factors are adhesins. Okay, adhesins means which has to be adapt for this one fixing. Okay, so what is the, this is very important, virulence factor for entamoeba histolytica. They might come as a short notes also. So amoebic lectin antigen, this is called, it is a 2260 kilodalton galactose and N-acetyl galactosamine inhabitable surface protein. Two subunits in here, which is the heavy and which is the light. And the lectin antigen is the principal virulence factor present on the surface of trophozoids of pathogenic E. histolytica, but not on non-pathogenic this one E. dispar. Its various pathogenic mechanisms are adhesion. They will fix by binding to glycoprotein receptors on intestinal epithelium and vascular endothelium. Cytotoxicity by contact-dependent cytolysis of the target cell by increasing the calcium level, and complement resistant. Okay. Other adhesins include 220 kilodalton lectin, 112 kilodalton adhesin and surface lipophosphoglycan. Okay, so other virulence factors are amoeba 4, then cysteine proteases, hydrolytic enzymes, neuraminases, and metallocollagenases, and then thioredoxin reductase system. So this amoebic liver abscess, okay, it's a very emergency thing. Okay, it's a very it's emergency. I mean, so whenever a person came with the amoebic liver abscess, we have to directly start the drugs. Okay, we have to start as soon as possible. Okay, so amoebic liver abscess, they are mostly found in young adult male. Most common affected site is the posterior superior surface of the right lobe of liver. Okay, then anchovy sauce pus. We call this as the anchovy sauce pus. See, this is the liver. Posterior superior surface of the upper right lobe of the liver, and this is the pus, this is the abscess. Okay, this is the, which is called as the anchovy sauce pus. They used to ask mainly in the this thing, what do you say, this one, your multiple choice type. Okay, this is very important. Then clinical manifestation of the amoebiasis. So asymptomatic amoebiasis is 90%, means most of them will be asymptomatic. Okay, and then intestinal amoebiasis, it will give rise to amoebic dysentery, amoebic appendicitis, amoeboma, fulminant colitis. Okay, amoebic liver abscess. Okay, hepatomegaly and fever. Most consistent features: weight loss, sweating, and weakness. Then very rarely jaundice and cough. See, intestinal amoebiasis. Suppose if a person or if a patient came to you with the intestinal amoebiasis, what you have to do? First of all, you have to make sure whether it is intestinal amoebiasis or not. Okay, then after that you have to start the treatment. Then first of all, then in the meantime of the treatment, you have to do this stool microscopy. We have to do with the help of the weight mount. It can be either the iodine mount or it can be either the saline mount. Okay, permanent stent, which can be done, example, detect the cyst and trophozoid. Histology, like intestinal biopsy, then with the PAS or hematogenin use. Instead, they will refer the trophozoid. Trophozoid, that is the adult form. Stool culture, we can do polygenic and agenic culture. Then stool antigen detection, like copro antigen ELISA and all. Okay. Then serology, like this thing we can do amoebic antigen ELISA, 170 kilodalton of lectin, and amoebic antibody isoenzyme and all we can do also. Okay, this is the most important thing for the diagnosis, for in the microbiology point of view. So this is important. How can you see there is a differences in the stool character between the amoebic dysentery and the bacillary dysentery? Okay, because both of them are dysentery only. See, for amoebic, it is mostly caused by the parasite, and for bacillary, it is mostly caused by the bacteria. So By amoebic dysentery, by pathology, this is for the pathology point of view, I'll talk about our microbiology, that is the stool microscopic picture when we see, okay. Then the number of motion which will pass in amoeba is 6 to 10 per day. In case of bacillary, which has been caused by the bacteria, it will be more than 10 per day. Amount will be copious amount, it will be very large in amount, okay. Then in bacillary, it will be small quantity, color will be dark red, and in bacillary, it is bright red. Odor will be very offensive in case of amoeba because it's parasites now. And in case of the bacillary, it will be odorless. Okay. Reaction will be acidic, the other one will be alkaline. Consistency not adherent to the container and adherent to the container. Differentials in stool character between the amoebic dysentery and the bacillary dysentery in case of the microscopic feature. When we seal the microscope, okay, then red blood cells RBC. So RBC will be mainly in case of the amoebic dysentery as compared to the bacillary. In case of bacillary, it will be less. Cells will be few in case of amoeba. In case of bacillary, there will be numerous. Macrophages, like few will be there, then numerous will be there. You charcoal latent crystal. This is the diagnostic feature of the this one amoebic dysentery. Okay, present in the amoebic dysentery and absent in the bacillary dysentery. Pycnosis body means the body which doesn't have any regular shape and sizes. Okay, this is called as a pycnotic body. They are present in the amoebic and absent in the bacillary. Okay. And organism detected amoebic dysentery will detect entamoeba, histological, or trophozoid. In case of bacillary dysentery with bacteria, which is the Shigella or Salmonella. So, laboratory diagnosis of intestinal amoebiasis. How can you diagnose this is the intestinal amoebiasis? First of all, we have to collect the sample. Okay, then we have to do the stool microscopy. We have to see. Then after the microscopy, we have a microscope. Sample collection. First of all, how do you collect the sample? Well, for collection of the sample, we require a clean mouth bottle. Okay, clean mouth bottle we require. Then after that, we have to tell the patient to collect their stool. Okay. Then after collection of the stool, then we have to close it very nicely. Then after that, we have to take it to the laboratory. But the ideal time is we have to see it within like this one within 30 minutes. Okay. Then stool microscopy means which we can see with our naked eye. There is the microscopic pictures. Okay, like suppose whether it is offensive in color, whether it is offensive in smell, then after that whether there is any blood, if from a blood is there or not, that is a microscopic picture. The stool microscopy means we have to see we have our own microscope. Trophozoid quadrinucleated cyst. Quadrinucleated cyst means this one, which composed of the four this one cysts. Okay. Then see, this is the microscopic picture of the cyst of entamoeba histologica. See, this is the saline mound. Saline mound, okay. See, this is the cyst. 1, 2, binucleated, okay. This is the iodine mound, usually yellow in color. 1, 2, see. This is the binucleate. And this is the trichome stain. See, 1, 2, see. This is the cyst. Not the chromatic body with blunt end. This is the chromatic body, and this is the chromatic body. Okay. Trophozoid and the cyst of entamoeba histologica in saline mound and iodine mound. In saline mound, what is the trophozoid? That is the adult form. Demonstration of the hematophagous trophozoid in the freshly passed stool is considered as a gold standard microscopic test that measures 15 to 20 micrometer, actively motile with finger-like pseudopodia. Presence of ingested RBC. Erythrophagocytosis differentiate it from the entamoeba dispar. Single nucleus not visible clearly, but a fan outline may be detected. In case of iodine mound, which is yellow in color, though trophozoid can be stained, one should not report trophozoid based on iodine mound, as iodine kills the trophozoid and mortality cannot be appreciated. Okay. C. Cyst has a smooth and thin cyst, while immature cysts contain round refractile chromatic bars, useful to study the nuclear characteristic in case of the iodine mound. Cysts appear round, 12 to 50 micrometer in size, then containing 1 to 4 nuclei. Nucleus has a central karyosome and fine peripheral chromatids lining the nucleus membrane. Both appear bright yellow. Cytoplasm appears smooth and hyaline. Okay. Glycogen mass, that is destined golden brown. See, we already know this iodine mound will be yellowish in color only. Okay. Chromatic bars are not stained in iodine mound, but stain can be but it can be stained with Barrow stain, surgeon stain, and the acrogen orange stain. Stool culture. Where do you do the stool culture? What is the media? It is the National Institute of Health NIH media, Voig and Derbola X serum medium containing the log solution. Stool antigen detection. That is the antigen antibody detection. You can do it with the help of the ELISA, detecting 170 kilodalton of lactin antigen immunochromatographic test. Serology we can do with the help of the amoebic antigen ELISA is then using monoclonal antibody specific for 170 kilodalton of lectin antigen, serine-rich entamoeba histolytica protein SREHP, which is called as the lysine-rich surface antigen. Amoebic antibody ELISA, IFA, and the IHA. ELISA means enzyme-linked immunosorbent assay, and IFA means this one fluorescent assay, and IHA means hematographic assay. Okay, then exoenzyme gymodyne analysis, molecular diagnosis, which we can diagnose the molecular form. Obviously molecular hoga to we have to know there should be PCR, nested multiplex PCR, which will target the small subunit small rRNA. By small rRNA we mean the ribosomal rRNA. Okay, genes that can differentiate, that is the entamoeba histolytica, entamoeba dispar, and entamoeba moskowski. Real-time PCR, which is called as the Tegman, targeting a gene S, that is the small rRNA gene. Biofire film array. For your exam point of view, this laboratory diagnosis, okay, microbiology for laboratory diagnosis very important. Okay, imaging method, which we can do it with the help of the colonoscopy, can be performed to detect the collar button or flask-shaped amoebic ulcer. Okay, this colonoscopy. Okay, other non-specific findings like charcoal-added crystal, which is found in the amoebic dysentery, but not found in the maxillary one. Moderate leukocytosis in the blood. See, amoebic liver abscess. Okay, how to detect the amoebic liver abscess? Amoebic liver abscess means, remember that entropy, source, pus, and all. Okay, so there will be a suppose a patient with the amoebic liver abscess, they came to you. Okay, they came to you like this one, sister, I am having pain in the upper right superior quadrant and all, what to do. Okay, so you can't just directly say, okay, this that the patient is having, because amoebic liver abscess, you might get confused with the appendicitis also, because appendix is also in the upper, there in the right part, in the right side of the abdomen. So for this reason, first of all, what you have to do, you have to make sure whether this is the amoebic liver abscess or the appendicitis. Okay, yes, in order to subdue the pain, you can give some some anti this one some this one, what to say, drugs and all. Okay, in order to decrease the pain. Okay, so for that reason, then after that, then what you have to do, you have to diagnose it. First of all, microscopy, they will detect the trophozoid, that is the trophozoid means the adult form. Then antigen detection by ELISA. Then antibody detections, like we already know, ELISA, IFA, IHA, and all. Okay, then molecular diagnosis like nested multiplex PCR and real-time PCR. Okay, PCR means polymerase chain reaction. Okay, we can diagnose it. Then ultrasonography, detect the site of the abscess and its extension. We have to know how much collection of the abscess is there in the right upper, in the liver, how much they have this one transgressed and all. Okay, we have to know. This is the ultrasonographical importance. Then see, suppose if a patient comes to you with this thing, pain in and all. Okay, so what will you do? So for the initial period, you can give this pain relief and all. Okay, but the thing is that proper drug therapy we have to give now. So for that, it's divided. This is important, like luminal agents. Okay, luminal agents, they act on the amoeba present in the gut lumen. Lumen means our intestine. Okay, examples include iodoquinol, paromomycin, dalogenate furoate. Tissue agent like effective on the intestinal wall, liver, and other tissue. Examples include metronidazole, tinidazole, ronidazole, benzimidazole, imitin, chloroquine, and all. Okay. Then asymptomatic carriage, like those people who doesn't present with any signs and symptoms. Okay, for that reason, for those people, what we have to give the luminal agents like iodoquinol, then uske baad paromomycin. Then acute colitis, we have to give these two agents like metronidazole, tinidazole, and luminal agents. Okay. Then amoebic liver abscess, what we have to give tissue agents like metronidazole, which is 750 milligram, or IV for 5 to 10 days. Then tinidazole and all. But luminal agents also. Luminal agents like iodoquinol, paromomycin, and dalogenate furoate. We have to give this one also. Okay. Then I think I have forgotten something. Okay. This one. We have to know about this life cycle, no? Okay. See, mode of transmission. This is the life cycle. Life cycle of Entamoeba histolytica. See, this is the liver abscess. This is the lung abscess. This is the skin ulcer. This is the brain abscess. Okay. This is the extraintestinal amoebiasis. Amoebiasis can be extraintestinal or the intraintestinal. Extraintestinal means which takes place this one outside the intestine, and intraintestinal means which takes place inside the intestine. Okay. These are the intraintestinal. Okay, extraintestinal means it can be caused in the liver like that of the amoebic liver abscess. It can be caused in the brain, but brain is very rare. Okay, then lung abscess and then skin ulcer. Okay, these are the extraintestinal. And in the intraintestinal, how it can be caused? They are mostly found in the lumen, like in the large intestine or in the small intestine. Okay. See, infective form is the quadrinuclear cyst. By what do you mean by quadrinuclear cyst? We have 1, 2, 3, 4, okay, which will be surrounded by the ectoplasm. There will be endoplasm, and there will be RBC, WBC, chromatic bodies, and all. Okay, this is the quadrinuclear cyst. Okay, so we ingest it, suppose by like fecal-oral route, we ingest it, ingest cardia. Okay, then after that it came to the small intestine. It came, and then 1, 2, 3, 4 still intact only. Then it came 1, 2, 3, 4, 1, 2, 3, 4. Okay, see. After that, what this is called? This is the metastasis. Okay, this is the metastasis. They are coming. Then after that, they will take out their this one ectoplasm and all. Okay. Then after that, they become naked. See, 1, 2, 3, 4. Okay. Okay. Then after that, see, there will be trophozoid formation will be there. See, trophozoid formation. By trophozoid formation, there will be RBC, WBC, everything will be there. Okay, they become mature. Okay, they become mature. This is the formation. Okay, then after that, once they go into the intestine, they might cause intestinal ulcer. Okay, this is the intestinal formation. They might cause intestinal ulcer. Okay, this that which we call as the flaccid ulcer and all. Okay, they might cause this one. See, they are coming other formation. Okay, this is the adult formation. From small intestine, they will go to the large intestine. This is the mature trophozoid. Then after this, what will they do? They will change into precyst. Okay, pseudopodia formation will be there. Okay, pseudopodia formation will be there. Then after that, they will give rise to the uninucleated cyst. Only one nucleus cyst is there. Okay, then after that, they will give rise to binucleus. Then after that, again, they will give rise to tetranucleus. And the cycle will continue like this. Okay, and so more and more amoeba will be like accumulated inside the inside the large intestine. In the small intestine, the first thing like those quadrinuclear cysts, and after that formation of the adult formation will be there in the small intestine. Then after that adult formation, they will give in the last intestine again, they will give rise to the cystic form again. Okay, this is the life cycle of the this thing, Entamoeba histolytica. See, uninucleated cysts, cysts in feces, seen in carrier and active infection. Once they become the quadrinuclear cysts, they can go directly either to the feces or somewhere. But mostly in the feces they used to go because it is completed in the last intestine. Okay. Okay. Then after that, this is very important for diagrammatic point of view, this pseudopodium and all, ectoplasm and all. Okay, they might ask you like give a schematic diagram of the life cycle of the Entamoeba histolytica. The life cycle of the Entamoeba histolytica, you can just draw like this only. Okay, then it can be understood only. And then this is the diagrammatic representation of the Entamoeba histolytica. Phylum taxonomy classification is not that important. Then amoebiasis. Then just know about this history of the Entamoeba histolytica. Entamoeba histolytica, who described it and when it was discovered. Okay. Then here in these drugs, I forgot I think to mention something about GR. Common area is going to be finished only. I have already finished only, but the thing is that I missed some point. That's why. Okay. Then common antiparasitic drugs. Here in the common antiparasitic drugs, this metronidazole, tinidazole, or nidazoles are important. Then which will be followed by amphotericin B is also important for this Naegleria fluoride. Okay. Then giardiasis, Dr. Sagar will take place I think for giardia, so he will explain there. And then trichomoniasis, which is the this one. Vaginalias, metronidazole or tinidazole. Okay, see trichomoniasis given to both the partner. Here, both the partners has to be given, both the male and the female. Okay, and then Chagas disease, not that. Then drugs for malaria is very important. And in the indirect cause or the complex life cycle, this is important. Plasmodium species, Echinococcus granulosus, Tinea solium, then Phaseolosis hepatica, Busky species and all. Okay. You guys understood now about this symbiosis, commensalism, parasitism, right?
Yes, ma'am.
Okay. So,
Yes, ma'am.
you guys have any doubt?
No, ma'am.
Any doubt? No, na?
No, ma'am.
Okay.
No, ma'am.
ठीक है तब तो. Okay. Then this is the class for today. Okay.
Thank you, ma'am.
Okay. Welcome.